It's my great pleasure to introduce to you Lars Klareskog, now as Crafoord Laureate and the recipient of the 2013 prize in polyarthritis. Lars Klareskog has made a series of striking and important observations throughout his long career as a rheumatoid arthritis geneticist. And these observations have, in a fundamental way, changed our understanding of the pathophysiology of this important and complex disease. His early studies on IAA antigens, their biochemical nature in the laboratory of Lars Rask, who I believe is here as well, set the stage for a whole field of studies subsequently addressing the importance of these alleles in autoimmune disease susceptibility and specifically rheumatoid arthritis. And I won't go into the many details of your biography, but you have served and acted as a champion in the field of molecular rheumatology for a long time, and in this way I think influenced the lives of many patients and improved their life expectancy and conditions in a major way. Now, the Crafoord Prize is usually awarded for molecular discoveries or often linked to dramatic changes in therapy. But the contributions of Lars Klareskog are not in the discovery of new genes or biomarkers. Citrullination, for example, as we know, was discovered by Van Venrooy and Serre, and the importance of smoking as an environmental risk factor has long been recognized. So Lars Klareskog stands out for the fundamental work he has performed in integrating novel molecular information with the clinical reality, and also for his crucial observation that genes and environment both influence the pathogenesis of this important disease in a fundamental way. And this was made possible, of course, by many aspects of your work, but it was made possible also by the assembly of patient materials and the vision of long-term follow-up and the search for genetic markers combined with careful characterization of the environments in which these patients live. So together with Dr. Winchester and Gregersen, you have allowed us to get closer to an answer, one of many maybe, to the question about the origins of pathology in rheumatoid arthritis. But there are many questions that I know that you will discuss. We know that identical twins don't always develop rheumatoid arthritis, so genetics is one factor. We know that we've learned that specific immunity is essential, but at the same time, blockers of innate immunity are currently the most efficient therapeutic tools that are available. And we don't understand what fits into Cinderella's slipper. And if I understood you right last night, you were going to give us some exciting novel clues as to what is going on. So I give you Lars Klareskog.
So thank you, Catharina, and also I want to convey my warm thanks to the Royal Academy of Sciences and to the Crafoord Foundation. And I must say one of the fantastic pleasures when Staffan Nordmark phoned me about this great prize was to share it with Peter Gregersen and Bob Winchester, who I've been working with and known for decades. I will make my presentation a little bit different, because I will go for what Claus Kjære said, that we are building on traditions. And my strong conviction is that science is a social business. You have to interact, you have to work with others, and then you could add some pieces yourself, but it always is a social business. So I will make it a little bit different, and also a little bit emphasis on my own Swedish background. So anyway, this is the result. It's beautiful and it's terrible, because this is how a cartilage may look like if we don't interfere with what is happening in rheumatoid arthritis. Painful, terrible, and beautiful. This is what we should avoid. As a matter of fact, this is one of my great heroes in medicine. As you see, he's not a molecular biologist. This is my teacher in social medicine and pediatrics in Uppsala University. And he showed that the main reason for severe handicap in kids, at least at that time, was accidents. So he went to the emergency ward and learned what was it that caused and interfered with the environment behind these accidents. Not only made his thesis and he had a number of students, but he also went to the insurance companies and to IKEA. And that is the reason why if you go to an IKEA shop, you see that the bookshelf has sort of a way of putting into the wall so that it doesn't fall on the kids. And so on. That changed the landscape in Sweden. And much more accident leading to handicap and death diminished drastically in Sweden, however, not elsewhere. And so he did, by just observing the environment, he changed condition and changed health. So I thought that would be a social medicine and public health scientist. That didn't happen, however, because it's people. And then I found this group, Per Peterson and Lars Rask. Lars is in the audience. They were fascinating, but they talked about molecular immunology and how you could sort of use chemistry in order to understand immunity. Quite different, huh? So Per and Lars had mapped the structure of MSC class 1 antigens just before Per came back from the United States and started new research groups in Uppsala. So I joined them with the purpose of isolating MSC class 2. Per told me it was easy. So I used my first 2 years of molecular sciences, totally artifacts. I usually tell this story to my students that they should just continue. Actually with the same project in my case. But then, of course, you can be encouraged a little bit by reading the literature. So I wrote, as I was to purify this first MSc class 2, I read this paper that came out some years ago from Peter Duarte, and that totally changed our understanding of the MHC, because it showed that the MHC somehow was involved in interaction between T cells and target cells. And Bob has given a beautiful encounter of what is the importance of that. So I thought that maybe we were working with an important molecule, but we didn't know how it looked like. So then we took and then came this observation, that maybe even it has something to do with rheumatism. We didn't know how. And again, I'm grateful for Bob's history. So finally, after these years of frustration, we managed to find a way of isolating these molecules. So we isolated these MSC class 2 molecules. These are the two bands. And you could see that, this one? Yeah. So this was the two bands, and that much later on was shown to be these two immunoglobulin-like molecules forming this molecule. So then you could make antibodies, and together with this guy who, actually, this is serendipity, always, because Urban Forsum, he knew how to make immunofluorescence on the skin. So we, of course, put our antibodies on the skin because that was what Urban knew. And then we came up with some peculiar cells there. We have no idea what it was. So I read the literature, and that was sort of 100 years of research on Langerhans cells, and suddenly we could begin to understand a little bit of what it meant. And that was, then I was captured in science. That was it. So then, why rheumatology? These two guys said that rheumatology research in Sweden was not very hot, I would say, at the time. This said that maybe you should not institute new professorships, but you should have some new scientists coming into the field from basic sciences. So that was what they said, and so I got a position to do rheumatology research due to these guys. So then I put these antibodies onto synovial tissues, and then this was normal. Lots of these peculiar cells in the synovium. So then you could, of course, postulate that something happened, that these molecules were involved in this type of activation of cells. Bob has worked that out as well. So then that was how it was captured into rheumatology. So the obvious questions coming from those, which are most of them still not answered 30 years later, so we have to go on for some more time. Then which immune reactions are acted? Why? Which cause disease? How do these interact? And how can we interfere? Okay? So actually, then I went to Hans Wicksell. That was an experience. And some of the ideas were fantastic and some were absolutely not possible at all. So I made both of them. And then Richard Holmdahl joined me and Hans and worked together. And I think that this was a very important lesson because from the original work on collagen arthritis and from many other arthritis, it was shown that there were many ways of getting arthritis. But when you knew the precise physiology of how you get arthritis in one of them or the other, then you could cure these mice. But they were very different ways of curing these different mice. So we had to know which were the genes, which were the environment, which was the precise immune reactions in the different ways, different strains getting arthritis. So that means that if we had rodents with defined genes, triggers, causative immune reactions, then treatment was in principle possible, but we lacked this understanding in human beings. So then came the breakthrough, which I've heard about, that this really, together with this crystal structure, put a completely new understanding of what things were. So with those of us who were extremely encouraged, and I sort of jumped on this paper, of course, as I did for the Björkman and Stroming and Don Wiley paper just earlier. However, scientific traditions, and this was already covered by Bob, and we did this very independent of each other, I would say. This is that from Uppsala University, obviously, Arne Theselius was mentor of Henrik Kunkel, and this is one of their papers. As you heard, Bob worked with Peter, but that was not the end of it, because also Arne Theselius happened to teach my predecessor, Börje Ordhagen, who made also a study on electrophoretic analysis, because Nana Schwartz asked her student, Börje, to work with electrophoresis, being led by Arne Theselius. I guess that they didn't understand so much of it, but they contributed to rheumatoid factors. But anyway, that just shows that translational research, if we want to call it that, is not new. Okay? Then I went to rheumatology in Stockholm with lots of collaborators, which was wonderful. And that means that you could begin trying to do these things in the clinic. And then we know that clinics in rheumatology has, over those years, changed fantastically. To a large extent due to these previous Crafoord winners. And that is why it's so fantastic to stay here and be the successor of them, including Tiny and Mark being in the audience. So this has changed the rheumatology completely. We have all these drugs, but it has not cured and not prevented yet, although it has changed lives fantastically. So in order to both understand what happened with the drugs and what happened with the disease course, we began in a fantastic collaboration which has gone on now for more than 20 years, a way of collecting information about our clinics, about our patients in Swedish rheumatology. So together with my colleagues, Staffan Lindblad in particular, set up registers following patients over time, both those who are recently onset and those who are treated with all these drugs. These are the numbers treated by the drugs developed by Crafoord winners, I would say. Now, so this, of course, gave us an opportunity, and this was a particular pleasure, because then I could begin thinking a little bit of my background in public health. And one of the heroes, Richard Doll, of course. And then see, could it be combined? So then we asked that and that was Lars Alfredsson, who is also in the clinic, in the audience, we asked the very simple question, why do you get rheumatoid arthritis? And the simple answer is, ask the patient. So we asked the patients a number of questions. What happened? What did they experience before they got the disease? I think we had 150 questions that epidemiologists ask. And then we put that together with the genes. And this is one of the studies. And then, of course, and that is the study which we have done. We asked a lot of questions, we took material, and we have still ongoing. Genes. One of the things which genetics has done, not only in our field, is that it has forced collaboration between us. It's not, 40 is not enough yet now. So we have to put it together. So these are just a part of the networks. And then comes Peter and Robert, who has been absolutely essential to getting this sort of sometimes not so easy continent to work together. And we have done similar things in Europe and also to Asia, as I will come back to. This is one way of saying the same thing as Peter said. That was when the GWAS came. However, I always show that this is not the whole truth, because concerning p-values, this is a truncated value. If you really look at the whole truth, this is the truth. So this is the MHC, this is p1222, and here comes the rest. And that means that we should think of the rest, and we may learn fantastic things, but we thought that maybe we should focus on this and pay some attention to that. So that is what we've done. Immunity. Rheumatoid arthritis has been considered an autoimmune disease, and lots of immunities beginning rheumatoid factors have been described. And those are some of the immunities that have been around more recently, to citrinated and also otherwise changed antigens, and more will probably follow. So the question is, which of those are important? How could you sort of use the new genetics and understand what is more important, and do subgroups exist? So ACPAs, anti-citrinated protein antibodies, were described by some chemists, Walter van Werner and Guy Serre, in the end of the 90s. And this is the enzymatic reaction from arginine to citrulline, and then the structure of these antigens is these molecules are changed. It's very specific. In rheumatoid arthritis, you have these antibodies in 60% of cases. In normal individuals, less than 2%, and not in other immunities, not in other diseases. But the first extremely specific antibodies that came, first being used mainly as diagnostic tools, not so much trying to understand what happened. But what really opened up the thing was studies that we did back in 2003, together with Leonid Padyakov. This is just the published saying that the entire presence of the MSC that has been described so much was only present in the 60% of patients who had these antibodies, and not at all in those who did not have the antibodies. So then the whole thing that had been with the MHC for all these years was only present for 60%, and we had a marker of this. So this was really this was one of those moments that Klaus referred to when we saw this result. And then, as the entire study of ADA was sort of set up, was to try to understand how did genes and environment interact. So we had lots of data on environment. Actually, we didn't think so much of smoking first. We had other ideas. So we just used smoking as the first prototype, just to see how did it work. So we put smoking together with genes, with the MHC genes, and divided into these two subsets. First with rheumatoid factors, and then with the ACPAs. See what happened. And to the great surprise, this was the moment when we saw this again, that in the ACPA positives, which said that smoking was only risk factor there, but furthermore, if you had no shed epitope, there was very little risk of smoking. If you have one copy of shed epitope, we are not smoking, of increased risk, 2 copies there. But look, this was the fantastic synergy and interaction which gave this 20 times increased risk. Whereas for the ACPA negative disease, there was no risk, neither from smoking nor from the shared epitope. And that should give us a clue. Also, a student of Lars Klareskog's, he made this thing looking at the dose relationship, saying that indeed if you are a multi-smoker, then it's 40 times increased risk of having this disease. So we continue with interactions. We actually looked also on gene-gene interaction. This PTPN22 gene was the second one. Actually also was not a risk gene for all RA patients, only for the ACPA positive ones. And this is a collaboration with Peter and with our Dutch colleagues. So as you see here, the PTPN22 has quite little effect when shared epitope is not there. Has more there and a major effect there. No effect for the ACPA negatives. This shows that this gene, which influences the threshold of the B and T cells, only works here, so different mechanisms to be found there. And then you could continue. And so then one question was, is this, we heard beautifully from Bob, how the MHC genes differ in different parts of the world. And that is evolutionary type of thing. So of course we wondered, is this something that is only for the MHC genes that we have here in Sweden or in the United States? So we set up a collaboration with Malaysia. Also serendipity is meeting people. Has a funny story, but I will not take it here. So that means that they set up a study with lots of RA cases and asking exactly the same question, but a bit modified for a Malaysian context in Malaysia. And then, of course, they have different genes, they have different environments, and we saw what were the case in Malaysia. And that was, again, smoking is there, so there's the same type of thing despite different MHC genes, but women don't smoke in Malaysia, but they have anti-citrin antibodies. So, and there is the interaction with the shared epitope there as well. So there must be something else. So it's very clear that smoking happens to be the example of what can happen, but it's not it. It may be other things that happen, say, in Malaysia, that we still have to find out, and which may be with airways. And this is just what I think how complex disease we look in the future. That is, here we have various genes, here we have various lifestyles, and of course that should be expanded into all those genes that Peter now recognizing. And we could have a number of things. We will hear something about diet, salt, and other things in the afternoon. Should come here. So this is ACPA negative risk, lifetime risk. This is ACPA positive. So you see that if you have a certain genetic setup, irrespective of what you do, you get a disease. If you have another one, you will not get this particular disease. But if you have this, which is the most common variant of genes, then it matters a lot what you do. And this people should know. And of course, it's a great thing to try to understand, of course, which are the mechanisms. But here, both in Malaysia and elsewhere in the world, there are two major subsets of rheumatoid arthritis. Now, this was the obvious question. What is the mechanisms behind this? Because now we have this genetic epidemiology, and we could try to understand how does it happen. So the obvious thing was saying that smoking, silica dust, coal dust, whatever, were risk factors. They did something in the lungs. So we went back and looked for what happened in the lungs. And this showed that indeed we have citrination, and this is cells coming up from the bronchoalveolar lavage, putting some saline into the lungs and coughing. And then you get up these cells and see that you have citrination there. Also increased expression of some of the enzymes, peptide arginine deaminases, which cause the transformation of arginine into citrulline. That happened probably by the toll-like receptors. So this was the idea that we and others formulated based on that. So this is just that we think that smoking and a number of other things may change things in the lungs. And if you then have the proper, this was the idea of the Cinderella shoes, if you have certain MHCs, citrinated antigens may put, or other antigens may fit in there, give rise to immune response, and then we have these antibodies that occur before onset of disease. And some by some mysterious way, they would somehow get disease. So we had at least some clues of how that might happen. Now, this is not the only way. This is one of the several other possibilities that exist. This is periodontitis bacteria postulated by Patrick Venables from the Karolinska Institutet and Karin Lundberg when she worked with him. And this is a molecular mimicry that these bugs can also give rise to citronation. And there may be other ways as well. But anyway, if we go for the lungs, we try to see what happens then in the lungs. Maybe something there happens before you have the onset of all these antibodies. So together with Anka Katarina and Gudrun Rinnersdotter in our group, we set up a study, and our pulmonologist looking into the lungs. And this is first time a patient with rheumatoid arthritis sees a doctor, just often only a month or so after they have had the first symptoms. And then you see in those individuals having RA, an ACPA-positive RA, but whether they are smokers or not, they have these lymphoid follicles being formed in the lungs, which does not exist in neither in healthy smokers nor in non-smokers, and very little in the ACPA negatives. So something happens in the lungs. And when Anka and Gudrun measured the antibody titers against ACPAs, this is that we have higher titers of these antibodies in the bronchoalveolar lavage fluid than in serum, indicating that there is early on a local production there. So of course, why? I mean, you could understand what that happens in the lungs, but why do it somehow end up in the joints? And I must say that we still don't know for certain whether this is the immunity that really causes the diseases. So far, associations. But we could try to find out. So then some experiments done with our mass spec people here at Karolinska as well is that we try to identify whether the same peptides are present in lungs and joints. Some of them are. For example, from vimentin, we have found the same peptides being seen in joints as in early lungs. So we are selecting some of these peptides which are seen there and see whether you have antibodies towards them. So that means that now we have an idea what we should look for, and we should go into the molecular details. So then I come with the same picture as Peter has already showed you. This is dense mapping over the MHC molecule, over the MHC and then it's shown, Peter has already shown that you see now we have the shared epitope, but also some amino acids there that independently are associated with RA. And so for HLA-B, the MHC class 1, and for DP, another MHC class 2. I think it's obvious to think that there must, if all these independently are associated with this disease, it must be that several different peptides might fit in there, driving several different immune reactions that may contribute to disease. It becomes even more complex. So this means that we are now looking at a number of different citronated molecules and also other modifications, I would say, that sort of may fit in. So we looked at all these different antibodies. We made a rainbow. This is the more complex pattern of autoantibodies against the number of different peptides that you find, or proteins that you find in the ACPA-positive disease. And we made an assay for that in order to quantify this. It's just technology. I will not try this so that you could measure many of them at the same time. And this was, for me, also another very important thing, because now we have looked for not for the all anti-citrullinate antibodies, but for specific ones against enolase, but not against other antigens, against vimentin, but not against the other, against both vimentin and enolase. And you say that the relationship odds ratio for the shared epitope, if you have both these antibodies, extremely high. If you have only this one against fibrinogen, quite low. So this means that different immunities, and that goes for what Peter would call endophenotype. That means that now we have an endophenotype, which is a specific immunity, which is more related to the particular shared epitope which we are so interested in than the actual disease. But maybe these other DPs and the other things in the bottom of the grooves, maybe those are the ones which are governing this immunity. And this is something that we don't know. We are trying to find out in an ongoing collaboration between us. Okay? This is a very important study being initiated some 10 years ago by Solbrit pair in northern Sweden. She is also in the audience. And she took the biobank in northern Sweden the same time they did the same thing with blood banks in the Netherlands, and found that samples taken from these patients, these individuals who were healthy at that time, but later got rheumatoid arthritis, they had these antibodies many years before, up to 20 years before. And then you see these different antibodies coming up. So these are individuals becoming positive for these specific antibodies, and then for other antibodies. And of course, those which increase very rapidly before the onset of disease, which is here. Those are particularly interested, of course, in order to try what do they do. Is these the ones who trigger the autoimmunity and goes into autoimmune disease? Give some clues. So then, for more clues, we of course should go back into the synovium and trying to see what happens there. And are there antibodies being secreted there? There was, from before, evidence that there is indeed the local antibody production against citrullinated antigens, of course rheumatoid factors that has been known for ages in the joint. So then we used technology originally developed by Michel Nussenzweig's lab in Rockefeller to make cloning from single B cells. That work was done by Khaled Amara, postdoc in a lab together with Vivi Malmström's group. So he cloned these single B cells and then looked for the monoclonals that came out. And to our surprise, between 20 and 40% of all B cells, unselected, which were present in these joints, produced antibodies against these citrullinated antigens. You see here, and we have two more now having the same type of pattern, between 25, 22, 33% of all B cells produce these antibodies. And when they tested them in the ELISAs, you see, for example, for the inulase, these are the ELISA curves for antibodies against these ones from the different monoclonals, absolutely flat for the arginine. So they are very specific for the citrullinated antigens. And then, of course, if you have that many which appear in the rheumatoid joint, and if you look for antibodies against tetanus, for example, they're exactly the same between aqua positives and aqua negatives. So this is very specific for aqua positives. So then what we have heard, the whole story from Peter and from Bob, is that T cells are the ones which are influenced by the shared epitope and by the MSCs. So of course we should go back there, but now we have some idea to look for T cells which may help to B cells to produce these antibodies. And there are different ways of looking for that, and you could, for example, look for binding of various peptides to these T cells. But now we have some candidates. So one of the candidates was this inulase, which is one of several. So we took overlapping peptides from this inulase and saw whether it bound to 401, to 101, or to 404, the one different variants. And these are the bindings of the arginine variants to this particular one. These are the bindings of the citrullinated variants. In this case, they only bound to 401, but not to the other two. Whereas for vimentin, for example, it's different. So you find that you have different binding properties to various types of MSCs. And that gives you some clues of how to proceed. So then, of course, the further one is, again, with Vivi Malmström's work, taking T cells and then seeing, do they react with these candidate peptides? And these are, in this case, one of the citrullinated inulase. And for those who are not used to the FACS curves, these are dots are single cells, in this case expressing interferon gamma, in this case expressing anti-interleukin 17. And you see that if they are unstimulated, no such cells. If they are stimulated, some with this particular peptide, but not then with the arginine ones, then you get this interferon gamma production or IL-17 production if you have an hemagglutinin from influenza, then you have mainly the interferon gamma being produced. So these are reactivities that we can see. Then you could see how many are they. There is a technology which has also been around for some years where you put the peptide into the MSC class 2. I mean, now we have candidate peptides to put there. But the binding between this complex MSC, this is the MSC and the T-cell receptor, is normally quite weak. So you increase the binding by having multimerization. For example, tetramers, but you could also make more. And I'm sure that we'll have much more about these things from Kai Wucherpenning and others in the afternoon. So anyway, you could take these and see what T-cells are there. In this case, this is a collaboration with Benaroya Institute in Seattle, Jane Buckner and Jerry Nippon. And they've seen that in this case for Vimentin tetramers are binding much more, in this case to RS subjects, than they are to healthy controls, which are matched for the same MSC. So something happens with the T cells. However, they are not many. They are rather rare. So this goes with many other studies, but it means that we are not sure that these are the guys which are doing the most important thing, but it's a lead. It's a way of beginning. And then what we've done, and this is very recent data and published by, also with the collaboration with Benaroya, is that if you look for memory phenotype and for an Th1 bias phenotype, you see that the cells binding the tetramers from the RS subject are memory cells and are differentiated much more than the fewer cells from the controls. So you could begin looking at these things. So in essence, you could say that we are beginning, I would say, with the help of taking clues from the genetics, which is the basis of the whole thing, but also taking the clues from this environmental type of thing, what happens in the joints, in the lungs, citronation. You could have clues what happens in this immunological synapse. And you could also have clues to see what happens down there. Of course, now you've only seen what is there. Are these things really causing arthritis? Still not really answered, but well, I should say this as well. This is the recent data. So this is together with the crystallographers and the people who know about peptides. We have now taken the arginine peptides. This is from one of the enolase peptides which really have a T-cell response against. This is the arginine variant, this is citrulline variant. And by crystallography, now they fit very nicely into this pocket. And we really have T-cells in RA patients which recognizes these things in the context of our HLR 0401. So I would say that this thing that we postulated some 30 years ago, it took some years to have the first sort of model which we now can use in order to see are these T cells there and what do they do and do they help the B cells that it's actually producing. These are still open questions, but they could be addressed with methods that we have. Now, final thing is just to say that these antibodies, do they have any functional effects at all? And I would say that the most convincing thing has been done by Richard Hondahl's group, taking, no, this is just to say that the monoclonals that we have, they indeed stain the synovial tissue. They react with, there is citronation there in the normal, in the inflamed, but not in the normal joint. But some of the antibodies are, as a matter of fact, again reactive as citronated collagen type 2. And these particular antibodies, when made monoclonals in Richard's lab in the mouse, caused experimental arthritis. This is a mixture of such antibodies, and they cause arthritis. So some of the antibodies that are there can definitely cause arthritis. But again, these antibodies are not there in all patients, and not even in the majority in some. So there may be other mechanisms. And this was a very beautiful study that came a year ago with George Shett, and we contributed a little, that some of these antibodies may activate osteoclasts. And as a matter of fact, these antibodies bind to the surface of activated osteoclasts and directly activate them. And then if you add these antibodies in vivo, they can induce osteoclastogenesis, and then they can induce a osteopenia and also a joint destruction. And Anka Katarina's group has done all that also in vitro, so that in this case, this is an in vitro assay where you see how osteoclasts are activated by ACPAs. These are those big multinucleated cells. And you could see that in erosion assays, you have much more erosions there than you have in the control. And in the other non-ACPAs, because this is affinity purified antibodies, you could see that it's those antibodies that do those things, not other antibodies that are present in the sera of these patients. So this means that with the help of the beginning of the genetics, adding a little bit of genetic epidemiology, trying to add some mechanism of conventional immunology into the pot, you could see that these schemes are happening, bone marrow is involved, and you could begin to study these things in detail. And of course, that means that you could also begin to think about what to do about this longitudinal course, where you could, of course, work with primary prevention. You could begin to treat much earlier. That was what we heard about, what Peter and Bob mentioned. We could, of course, use the fantastic new treatments which we have there, better, hopefully, with knowing mechanisms. And of course, we could do these things. But in the end, what we really want to accomplish is, of course, using this knowledge about the MHC peptide, which is the basis of the prize which we are so happy to have, as a basis for new things. And then one example is, of course, our previous Nobel laureate, Ralph Steinman, who was one of those who showed that if you know what is the signal between the immunological synapse, you could both induce tolerance, but if it's activated and tuned otherwise from the environment, you get activated adaptive immunity. But you have, in order to sort of control this, you have to know both what is sort of determining this tonic and what are the specificities. And I think that we are approaching to begin, we are at the stage where we can begin to approach these things. And I therefore hope that what is possible, a little bit of history, could sort of encourage many people to go into this area, and that the three of us will continue exploring this also in the future. And with this, I want to thank my many collaborators over the world, and also again a thanks to the Royal Academy and to the Crafoord Foundation for this great honor of us. Thank you.